Go ahead, Ksenia.
Yeah. Okay. Hi. Thank you so much for meeting us here and allowing us to give a talk. It's our honor. We are a small company who are specializing in dealing with different data sets, which are usually not literally very nice and not very beautiful. They are completely fitted into the real human life, and we use different techniques. And usually we validate everything on a go because usually our clients provide data weekly or monthly. So this particular talk, we're going to focus on a cancer research. And this is like really very interesting because we have so much discovered. It was discovered different hallmarks and pathway and the receptor signal and DNA. But we literally have built all the targeted treatments only per particular issue. But we never looked at the cancer in another ways. So, and there is, for example, if we consider cancer, which is a solid tumors cancer, they are not responsible to many of those treatments. The cancer itself gets modified, it is getting resistant, and literally the patient relapses again. And this is the big problem within any cancer. Second problem within any cancer, nobody knows the originality of it. Why is it happening? If we have even this DNA or we don't have it, what is the real cause of it? It's not like we consume through the air transmission or bodily fluid or we add something. It's a very, very unknown origin why the cancer actually even happens. So, and the question which we're going to look at is, what is really the cancer? Can it be like a structural? Can it be a dynamical system within our human bodies? And how is it really behaving? And what is really the maybe driving cause? And if it is a structural, is it stable? Is it unstable? And what is it really possible at the end, hopefully, what is a possible targeted path for the treatment? So this is the particular talk which I'm going to give about, and the data was provided by the company as Kareem, and it is based on a National Institute of Health, National Cancer Institute, FDA, the current and recent trials going on, and questionnaires of the patients. So we looked, and the data set, as you can see, is quite pretty large. It was taken random, and we have been given this as to understand what is really we can discover just having this large data set. So we took the problem in a different ways. I'm not going to talk about much Bayesian causality. I'm going to talk about mixture of different methodologies and how you can mix and possibly look at this in a very different way. So the first things, what we did, we looked at the biological vocabulary as the main driving force, and we literally constructed a co-occurrent network and looked into Luvian community detection, which allowed us to discover different 13 coherent modules. And this we used as they have been like literally corresponding to biological, recognizable biology, receptor signal, mitochondrial repair, immune activation, and others. Then we aggregated and built Laplacian graph. And this comes from the theory of having a multi-species dimension, looking at the cancer cells when you don't know literally what type of cancer you have and what is originated. And this model allows you to absorb up to 25 parameters unknown. Then the second one, we looked into regulatory dynamic. And this is question which we ask a little bit different. Which of those 13 modules drives which? Not like what is leading and what is not leading, but which of those 13 defined modules drive and which. And this is a key analytical transport matrix for us, and meaning that the different layer inheriting the module basis exactly. So each of these matrices has own eigenvalues, and the most interesting thing that all of these matrices within the transport matrix have been literally stable. All of them lying in a negative real plane, which is remarkable, taking into account if you look per each kind of pathway. Then the third one, we took like a low dimension control layer, and we compressed the system into minimum effective aggression of freedom using lasso regression in an autoaggressive form. And we looked at each cluster, and eventually it all collapsed only into 2 clusters which have been dominant. So then what we did, we looked into the intersection of all those domains, and we combined all those 3 different layers in all possible ways. And we looked into the uniqueness, we looked into the actually eigenvalues or values which can be the same and which are unique per each of these intersections. So, and what we really figured out, that the first is instability of the whole total data set. And it is really impressive because we got like 28-dimensional operator driven by ordinary differential equations, and all of them are literally unstable with a value 3.13. And, but the interesting point, if you look at it, at each individual small matrix within the whole, this 28, it's literally stable. And this is really kind of maybe describing how the cancer is behaving when the targeted therapy is provided only per one of these 13 modules, which are responsible to biological responses. Second, we looked at we discovered 2 biological axes. The first is a tumor growth program. It's like receptor signaling, proliferation, surviving, and switching, and it is moving together as a control one system. Then another axis we discovered is a repair and a checkpoint. It's a central DNA repair, genome stability, and topologically, it is also like a growth program, and one dominates over another, which is also an interesting point. The third point we discovered is a survival mode versus death. So the first mitochondrial resilience is responsible, and it is supported eventually by the data which we got at the end, that if you have a mitochondrial survival, it literally feeds the cancer. But on another side, we got opposing the apoptic gate, which says that as soon as the cell is dying, cancer is dying as well. I mean, normal cell is dying, the cancer cell is dying as well. And this is really very interesting because this multidimensional matrix literally gave us the response, or that it is the DNA response and repair drug response is 29.5%, literally larger than a standard null. It is just literally not a simple correlation, it is a causality. We don't really cannot say which type of causality, but it is some type of causality. We think it is more Granger causality rather than Bayesian causality. And the last one is an interaction chain. We discovered that there is only one single path which was stable between all those 28 matrices. And the final word maybe for the, sorry, I was talking about it and forgot to switch. This is what we talked. And the last one is this is a really very interesting because it is validated by our clients, but on top of it, it is validated by a few publications of Hanahan, who is just recently proposed the research that cancer may be a dynamical system if we treat it and not targeted. And it was just published in 2026. We are a private company working for a client, so we patent everything. Don't do much publications, apologies, but yes, thank you so much.